afternoon. We're going to go ahead and begin. So we have been covering cells, cytology, and then we started talking about how do chemicals get in and out of a cell. So they get in or out of the cell through about 4 different ways, including diffusion, osmosis, active transport, and phagocytosis. So last time we talked about diffusion. And we said diffusion is simply where a chemical spontaneously starts to flow from an area of higher concentration to an area of lower concentration. Incidentally, all this stuff on cell membranes, diffusion, osmosis, active transport, phagocytosis, is in chapter 4, I believe, of your textbook. And we gave you 4 factors that affect the rate of diffusion, and those 4 factors were the difference in concentration between the inside and outside of the cell. The bigger the difference in concentration, the faster the rate of diffusion. Second factor was the size of the molecule. A smaller molecule goes into the cell or out of the cell at a faster rate than a larger one. In fact, if it's too large, it won't be able to get across the cell membrane at all. A third factor was whether the molecule is a fat or not a fat. So which did we say get through a cell membrane faster, a fat or something that's not a fat? A fat, because a cell membrane is fundamentally made up of phospholipids. It's fatty in nature. And the fourth factor was the temperature. The higher the temperature, the faster the rate of diffusion, which is written right there. So that's we covered all that last time. And so that takes us to osmosis on page E2. Now, osmosis is simply the diffusion of water through the cell membrane. We could just say diffusion, but they give it a special name, osmosis. So obviously, if a whole lot of water were to diffuse into the cell, it would cause a cell to possibly swell up and even burst. On the other hand, if a whole lot of water goes out of the cell, it would cause the cell to shrivel up, to get all shrunken, collapsed, and undergo what we call crenation. To help us visualize this, we made a note to see page E9. So we're going to look at page E9 to help us understand this concept of osmosis. Yeah, we have it on that page. All right, so right here, what I want to focus on is this drawing right here. Now you might say, whoa, your picture is a big mess. Give me a chance to copy it down. But we'll walk you through it as I always do. This is page E9. All right, so we're on page E9. In the middle of the page, the left-hand side. Now, what this picture shows, admittedly I've made it messy, is it shows a red blood cell. Let's zoom in on this red blood cell. And this red blood cell is smiling. See? It's got a big happy face. Now, we have learned that the cytoplasm, the jelly-like fluid inside of a cell, is 80% water. You'd say, when did we talk about that? This actually goes back to section D, actually section C. We have learned that a human is 60% water. The cells of our body are 80% water. So most of what's inside of a cell is water, but it's not 100% water. So I've represented the fact that there are other chemicals in the cytoplasm of the cell besides water by drawing some dots. Those dots represent proteins, sugars, vitamins and minerals. So those dots just represent whatever is inside that cytoplasm of the cell other than water. And then I kind of colored it blue in the background. Now, if we surround this cell, this red blood cell, which is a human cell, an animal type cell, if we surround this cell with a solution that is similarly 80% water and 20% other stuff, we would call that solution an isotonic solution or isotonic fluid. Have we ever seen that Greek root iso before? What does iso mean? Good, very good. It means same or equal. So we've seen that root appear in the word isotope and isomer So it keeps reappearing. It just means same, similar, equal. So in this case, what we're showing is that the fluid surrounding the cell has a similar, the same proportion, the similar concentration of water and other stuff outside the cell as inside the cell. And that means that since the concentration of water is on the outside of the cell is essentially the same as the concentration of water on the inside of the cell, water is neither going to rush into the cell, causing the cell to swell up and burst, nor is water going to rush into the cell, causing the cell to collapse. So if it flows in, so it's not, I'm sorry, if it flows out and collapse, thank you. So this is a happy cell. Now let's consider two other possibilities. What if we place this red blood cell, instead of surrounding it in an isotonic fluid, what if we placed it in what's called a hypotonic solution or hypotonic fluid? An example would be surrounding this red blood cell in distilled water. Distilled water means 100% water. So again, I've drawn this cell. Let's enlarge it. There's the cell. It's mostly water. It's 80% water on the inside, but it's not 100% water. We've drawn some dots to represent, again, the fact that there are proteins and sugars and vitamins and minerals and other chemicals besides water inside the cell. But this time we've surrounded the cell in pure water. I haven't drawn any dots on the outside of this cell. So it's 100% water on the outside. So is there a difference in concentration of water between the outside and inside of this cell? Yes, it's 100% water on the outside, it's 80% on the inside. There is a higher concentration of water on the outside of the cell than on the inside. So what is water going to do? It's going to flow in. And it's like any other chemical, it flows from an area of higher to an area of lower concentration. As the water flows into the cell, and I wrote here H2O is going in, here it shows H2O is going in. As more and more water flows into the cell down its concentration gradient, well, first of all, what do we call it whenever water diffuses either into or out of a cell? We call it osmosis, the diffusion of water. And as more and more water flows into the cell is going to start to swell up more and more and more, and it can only swell up so far before it bursts. And when it does, that's called lysis. We've seen that Greek root lysis before. It always means to break apart whenever it appears. So that's not a happy cell. In fact, that cell is a dead cell. Let's consider the third possibility. The third possibility, you'd say, whoa, what a mess. Okay, so let's see what I've drawn. Here's a cell. It's the same red blood cell. It starts out just like the other two cases. It's 80% water on the inside of the cell. So again, it's not 100% water. Again, I drew some dots to represent the fact that there are proteins and sugars, vitamins and minerals inside this cell. But this time we've surrounded the cell by what's called a hypertonic solution. A hypertonic solution, written right here. And here it says the cell was placed in a concentrated salt solution. Hypertonic means high in salt or high in solute, in sugar, in protein, but not high in water. It's high in anything other than water. So I've tried to represent this solution surrounding the cell. I wrote that it's just 20% water on the outside of the cell. That means it's, what's 100% minus 20%? What's 100 minus 20? 80. If it's 20% water on the outside, it's 80% salt on the outside, right? Something other than water. So I spent a lot of time drawing a lot of dots. You could have fun drawing lots of dots to indicate that while there is a lot of salt in this fluid surrounding the cell, there's not much water. So, is there a difference in concentration of water between the inside and outside of the cell? Yes, it's 80% water on the inside, it's only 20% water on the outside. So what's water going to tend to do? It'll flow out, flowing from an area of higher to an area of lower concentration of water. All right, and so this is again called osmosis, and as more and more water flows out of the cell is going to start to collapse because all the water is going out of it, and this is known as crenation. So the basic lesson we learn from this is that first, whenever water does flow either into a cell or out of a cell, it's called osmosis. Secondly, if too much water flows in or too much water flows out, we have cells that basically die. So it's an important subject. Now, there is a slight difference between what happens when animal cells, including human cells, are exposed to hypotonic solutions versus when plant cells are exposed to hypotonic solutions. And so what do you mean? Let's look at the picture right below. In the picture immediately below, on this same page E9, in this case it shows a plant cell. Now, we're going to learn more about plant cells. Some of you have already learned about animal and plant cells in your lab class. Plant cells are surrounding the cell membrane of plant cells is an outer rigid cell wall. And we've actually mentioned that plant cells have a rigid outer cell wall made up of a polysaccharide called cellulose. You might say, we never talked about that in this class. Yes, we did, on page D2, when we first explained what cellulose was. It's a polysaccharide found in plant cells. So, and we have already mentioned that bacterial cells have a rigid outer cell wall, not made up of cellulose, but made up of some other polysaccharide. So, because the plant cells are surrounded by a rigid outer wall, this is outside the cell membrane, so when you place a plant cell in pure water, 100% pure water, we wrote that the outer cell wall prevents the plant cell from bursting. So all that happens is that the plant cell becomes all plumped up with a lot of water, but it doesn't actually burst. So that is a difference between plant cells being surrounded by pure water versus animal cells, including human cells, being surrounded by pure water. In terms of surrounding plant cells with a hypertonic, a very salty solution though, it works just like with an animal cell. Again, if the inside of the plant cell was 80% water on the inside, if it was 80% water on the inside, but it was only 20% water on the outside, then water would flow out of the cell, water would flow out of the plant cell, and the plant cell kind of does start to collapse. It does start to collapse. So the main difference between animal cells and plant cells is that whereas animal cells will swell up and burst when exposed to water, a hypotonic solution, plant cells do not burst. They do swell up with water. Now, we can relate to this a little bit if we think of some stalks of celery. All right? So let's imagine we've got a stalk of celery, and we place the stalk of celery, let's say in a concentrated salty solution, a hypertonic solution. So what did we say is going to happen with the water in the plant cells if we surround them with a very salty solution? The water is going to flow out of the cells, because the inside the plant cell, it's 80% water, outside it's less than 80%, maybe 20% water. So the water flows out of the cell. Incidentally, one thing, a way of visually aiding you to understand this, basically water is going to tend to flow wherever there are more solute particles. Because wherever there's more solute or salt particles, there's less water. So water tends to be, in a sense, drawn towards wherever there's more solute or salt particles. So as the water flows out of the plant cells, the stalk of celery will start to wilt and droop, all right, because the water is going out of the plant cells, so they're kind of getting shriveled up a little bit. So it gets wilted. On the other hand, we could take this stalk of celery and revive it. We could make it look, return it back to normal. We could place it in pure water. And if we took that stalk of celery, celery, and placed it in pure water, so what would water start to do? It would start to flow into the plant cells, because it's only 80% water in the plant cells, and as the water flows into the plant cells, now remember, they're not going to burst, but they will get all plump, filled with water, and that'll cause the stalk of celery to become upright and crisp. So it'll be all crisp. Now, does this kind of explain why it is that in the produce section of a market, aren't they constantly spraying water on the lettuce and the celery? Because nobody wants to buy wilted lettuce and wilted celery. So by constantly spraying water on it, the water enters the plant cells and it keeps the leaves of lettuce and cabbage and celery firm and crisp rather than all wilted and limp. Incidentally, one other visual aid of this is if you pour salt on a snail. I'm always surprised by how many people have done that. But when you pour salt on a snail, so what did we say happens when you surround cells with a very salty environment? It, in a sense, draws the water out of it. So all that bubbling that you see when you pour salt on the snail is the water, in a sense, being drawn right out of the cells of the snail, and all the cells of the snail are collapsing, and the snail is just all kind of collapsing and imploding. That's what you're doing to the snail. I always point out, I myself would never put salt on a snail. I would never do that. I throw them in the street and watch cars run over them. What do you think that, you think that's more painful?
It's quicker.
It's quicker. Instantaneous death. Right? Try to watch the snail trying to get out of the way of the car, and it's kind of moving way too slow.
Experiment, your mother tell you to put salt on the snail, that's what it's going to do.
All right, so now you understand how the water is coming right out of the cells. All right, so let's summarize what we've been learning by returning back to E2. Returning back to E2. So we have been talking about osmosis. What is osmosis? It is water moving across the cell membrane of a cell. And we've seen that if too much water flows into a cell, at least animal cells, it'll swell up and undergo lysis, rupturing. In both plant and animal cells, surrounding the cell with a very salty solution, low in water, will cause the water to be drawn out of the cells, out of the animal cell, out of the plant cell, and the cells will shrivel up and undergo crenation. So neither one of those is very good. Okay, so we've been talking about, that was crenation. We've been talking about the movement of water in or out of cells. Now another way that chemicals go in and out is by something called active transport. We actually talked about this last time. We said that there are proteins embedded in the cell membrane. Last time we called these transporter or carrier proteins. And we said these transporter or carrier proteins transport specific chemicals through the membrane. Now, if it uses energy supplied by this gasoline called ATP, then we call this active transport. Let's remind you, we've seen a picture of this previously back on page E8. So let's look at page E8. We've seen it before, page E8. It's in the lower half of the page. And in the lower half of the page, so here it shows a cell membrane, right here. And it shows these proteins embedded in the cell membrane. And it shows some box-shaped chemical, some specific chemical, that attaches to this protein. And this protein, using energy supplied by ATP, forces this chemical across the cell membrane. This is known as active transport. Now, your books will also mention that, and we mentioned it last time, that sometimes a protein will transport a chemical across the cell membrane without requiring energy, and they give a different name to that. They call that a passive transport. But I mostly want to focus on active transport. Yep, question? No? Okay, so these are both described in the box here at the left. So let's give you a couple of examples. Returning back to page E2. Back on page E2. So as examples of active transport, we mention iodine uptake by cells of the thyroid gland. In other words, there are iodine ATP transport proteins. Iodine ATP transport proteins, or pumps. You'd say, what's that word say? Iodine. I-O-D-I-N-E, iodine. Now they're called iodine ATP transport proteins, or transport pumps is commonly how they're referred to. Now where are these proteins or pumps? They're found in only one cell in the human body. They are found in the cells of the thyroid gland. So I've drawn a thyroid gland cell. I've drawn a cell. I represented that there are these proteins, these transporter proteins embedded in the cell membranes of thyroid gland cells. And these proteins actively transport iodine into the cells using energy provided by ATP. Now, you might say, I'm very confused. I don't understand what you're talking about. I want to remind you of something we learned about way back in section C. Remember we said that you could take radioactive iodine, and if you swallowed it, where would eventually all the iodine normally end up? In your thyroid gland. Now the question, remember, and there was a video of a guy where he showed using a Geiger counter device how all the radioactive iodine, the radioactivity had concentrated in his thyroid gland. The question is, why? Well, how does all the iodine end up there? If you swallow this iodine, whether it's radioactive or not radioactive, why doesn't it just end up everywhere in your body? Why does it all end up being concentrated in the cells of your thyroid? And the answer is because only the cells of your thyroid gland have these proteins that use energy to force iodine into these cells. Because that iodine is used by these thyroid gland cells to make the hormone thyroxine. And so in fact, they commonly will give people radioactive iodine to see if their cells of their thyroid gland are working properly. Are they accumulating iodine like they should? Because if it's working right, all the iodine that you ingest should end up inside the cells of your thyroid gland. That's because of these active transport pumps. Now I want to give you another example of active transport. I actually gave 3 examples here. I've just talked about iodine uptake by cells of the thyroid gland. Glucose is actively transported from your intestine by glucose transport pumps. But I want to talk about what are called sodium-potassium ATP pumps. And our first thought is, what are sodium-potassium ATP pumps? These are actually found in all cells. Every single cell has this active transport protein. All cells. Now you might say, when you say all cells, do you mean like just all cells in a human? No, I actually mean all cells of every living organism. All cells in humans, all plant cells, all bacterial cells, all cells of fungi, all cells of every living thing. In the cell membranes of every living cell are proteins that actively transport sodium ions out of the cell and at the same time transport potassium ions into the cell. This is known as a sodium-potassium ATP pump. You'd say, can you write that down? That's what it says right here, sodium-potassium ATP pump. It uses ATP to provide the energy to pump or force sodium ions out of the cell and accumulate potassiums on the inside. Now, through a mechanism, without going into any more details about this transporter protein, it is by virtue of pumping sodium ions out of a cell and pumping potassium ions in that it causes there to be a difference in electrical polarity, or in other words, an electrical voltage in every single cell. Because this pumps actually more sodium ions out of the cell than it pumps potassium ions in, the outside of the cell, of every cell, ends up having a positive electrical polarity, and the inside ends up having a negative electrical polarity. So every single cell of every single living organism has an electrical voltage, or an electrical polarity. This difference in electrical polarity between the inside and outside of the cell, such that the inside is negative and the outside is positive, is called an electrical voltage. Now we might ask, okay, wonderful, how big is this electrical voltage? The electrical voltage of every cell in your body and every other living thing is approximately 1/10 of a volt. 1/10 of a volt. Let's see if we can relate to that a little bit. The most common batteries that are used are like AA or AAA batteries. Right? Has everybody got, some electronic device that uses a AA or AAA? And even if we could even talk about flashlight batteries that are size D or C, because they're all the same voltage. Does anybody know what a AA or AAA or size C or D battery is, what the voltage is? Anybody know what it is off the top of their head? You can look it up. Okay, we have somebody checking it out right now.
1.5.
Thank you, it's 1.5 volts. They're all 1.5 volts, all of them. All right, thank you. So a AA, a AAA, a size C, a size D, they're all 1.5 volts. All right? So I just said a cell is 1/10 of a volt. So it's like 1/10, really less than 1/10 of what the voltage is of most of the commonly used batteries. Not very impressive, right? Now multiply that times the number of cells in your body. You have 60 trillion cells. So the amount of electrical energy in your body is awesome. This electrical energy, every single cell is like a battery of about 1/10 of a volt. And this electrical energy is the basis of the electrical activity of our nervous system, the electrical activity of our heart, the electrical activity of our muscles. All of these parts of our body generate electrical currents. And it's all related to this sodium and potassium. For those of you who've had a psych class, right, those of you who had psychology, they might have covered a little bit about the action potential in a nerve cell and how it involved something having to do with sodium ions flowing in. Anybody remember that? Something about sodium ions flowing in and potassium ions flowing out. So it's all related to sodium and potassium that creates this electrical voltage. Now, just as another way of relating to this, if you ever saw that any of the 3 parts of the Matrix movies, remember the Matrix movies? The main lesson of the Matrix movies is that you should wear a black trench coat and sunglasses to look cool and be able to dodge bullets. All right? But if you saw any of those movies, you'll remember that humans were actually being kept in incubators by the machines. The machines had taken over the world, right? And what were the machines using humans for? As a source of energy, as batteries. All good science fiction is based on some real science. And in fact, every cell in your body is a battery, and you have awesome electrical energy. And so in the movie, obviously it wasn't true, but the machines were using humans as batteries to power the machines. That was the whole premise of the movie. That was the part I understood. Parts of it I had no idea what was going on. All right, now, the amount of electrical energy in our body is really, truly awesome. And it's all related to this sodium-potassium ATP pump, which is found in every living cell of every living thing. So even plant cells have a voltage, as even bacterial cells have a voltage. All right, we've been talking about using energy in the form of ATP to force chemicals either into a cell or out of a cell. Iodine is forced in, sodium ions are forced out, and so on. Now, 2 other ways that things can get in or out of a cell. Number 4 and 5, phagocytosis, which is also known as endocytosis, and secretion, which is also known as exocytosis. Now, I did make a note that these processes also require energy provided by ATP. To understand these two, let's look at page E7. So if we turn to page E7, on page E7, we're going to look on the lower right, on page E7. And on the lower right, it's labeled phagocytosis or endocytosis. Now, the word phago means to eat, and cyto means cell. So the cell is, in a sense, eating. What this picture shows is actually 3 images of a white blood cell, a WBC. And these little guys just outside that white blood cell represent bacteria. We know bacteria are much smaller than human cells because they are prokaryotic cells. The bacteria are the smallest living cells. And white blood cells are more like typical human cells in size. Now we already know, we've already learned how a white blood cell can recognize that bacteria aren't us, because they don't have the correct identification markers or recognition site glycoproteins on their outer surface. So we know that the white blood cells are going to swallow up and destroy these bacteria. How does it swallow the bacteria up? You'll notice in the middle picture, you can see the cell membrane starting to fold inwards. And so it's starting to, in a sense, swallow up. And by the time we get to the third picture, 3 of the bacteria have been swallowed up by the white blood cell and now are contained or enclosed within what's called a food vacuole. So now this requires ATP in order for this cell to change its shape. This is called cell eating or phagocytosis, phago means to eat, or endocytosis. The root endo means inside. The cell is taking inside something that was on the outside, it is taking in. Now the very opposite of this, the very opposite process is shown at the bottom picture. In the very bottom picture, it shows what's called secretion or exocytosis. What's that? In this picture, it shows that there's these little containers or vesicles inside the cell containing a chemical. These vesicles move towards the cell membrane. They unite or fuse with the cell membrane and release their contents to the outside of the cell. This process of releasing something that was on the inside of the cell and releasing it to the outside is known as secretion or exocytosis. The Greek root exo, as in the word external, to the outside, as opposed to endocytosis, bringing it to the inside. Just like endocytosis or phagocytosis, secretion or exocytosis similarly requires energy in the form of ATP. So, let's summarize how things go in and out of a cell by once again looking at page E8. So on page E8, this is page E8. So this really summarizes. We know that there are ion channels where these electrically charged atoms, which are extremely small, can diffuse in or out of the cell. We know that fat-soluble chemicals, things that are fat, can easily diffuse right across the fatty membrane. All of this is described right here in the box at the left. We know that water can get across cell membranes, and we know that that's called osmosis. If too much water flows into the cell, it may swell up, and in the case of animal cells, even burst. If too much water goes out of the cell will collapse or undergo crenation. We've talked about active transport. Active transport is when energy provided by ATP is used to force chemicals in or out of the cell. We gave the example of iodine, ATP pumps that pump iodine into thyroid cells. We gave the example of sodium-potassium ATP pumps, where sodium ions are pumped out and potassium ions are pumped in. And that creates an electrical voltage across cells. And what it shows here at the bottom is how the cell membrane can change shape. And that's what happens in both phagocytosis and secretion, where the cell membrane changes shape, and again, that requires energy in the form of ATP. So we have been describing or summarizing some of the main ways that chemicals, substances, can be taken into the cell or be expelled out of the cell. Let's go back to page E2. Back on page E2. So now we want to talk about the cytoplasm of the cell. Now, we've already learned several times now that the cytoplasm inside the cell is mostly water. It's about 80% water. 80% water. Now, after water, the next major chemical that makes up cytoplasm are proteins. So in other words, cytoplasm is mostly, as far as composition, it's mostly water and protein. Incidentally, anything that is made up mostly of water and protein will have the consistency of Jell-O. So if you were to make Jell-O, the way you actually make Jell-O is it's actually made up of a protein, proteins from plants usually, and you mix it with water, usually have to heat it up, and you just mix the water with the protein and Jell-O. It's called gelatin, and gelatin protein, and when it cools down, it has the consistency of Jell-O. Now, let's talk about cooking right now. I know that's why you're taking this biology class. We'll talk about cooking. So the weather's a little bit on the cold side, so let's say we all make soup tonight. All right, I don't care if you make chicken soup or some other meat soup, let's make soup. All right, so we're going to make it simple. We're going to take a pot. We're going to throw some meat in the pot. And we're going to fill the pot up with water. Pretty simple. All right, and then we're going to boil the hell out of it for several hours. Now, as we boil the meat, all right, what is meat? Meat is actually muscle cells. It's muscle tissue from an animal. Think about that when you eat it. So you're eating muscle cells. Now, what's happening, muscle cells are very high in protein. There's a lot of protein in all muscle cells, including yours. As you boil the meat, that muscle tissue, some of the proteins in the muscle cells are actually coming out of the muscle cells, the meat, and dissolving in the water. So after we boil this thing for a couple of hours, let's let it cool down, and let's put it in the refrigerator overnight. So when you pull it out of the refrigerator the next morning, there's some fat on the surface, because fat won't mix with the rest of it. And after you remove the fat, what does the rest of it look like? Like Jell-O. So what do we know it's mostly made of? Water and protein. That's, anything made out of water and protein is gelatinous. So some of that protein that was in the meat has now dissolved in the water, the soup. All right, so clearly it came from the muscle cells, so there's a lot of protein. Now, many years ago, they used to think these proteins were just kind of floating around in the cytoplasm. But we now understand that these proteins are actually organized into a complex pattern. This is known as a cytoskeleton. It is a network of proteins, right? So there's really a whole network or pattern of proteins. So they're not just floating around, they are organized into what's called a cytoskeleton. Just as we have a skeleton that gives us form and shape in the form of our bones, our bony skeleton, the proteins in the cytoplasm are what give it shape and form. Now some of these proteins, in the cytoplasm are described as microtubules, and others are described as microfilaments. What's the difference? A microtubule is just like its name says. It's tube-shaped. It's actually hollow in the center. So it looks like a pipe or a tube. Other proteins are thinner, and they're more like a solid wire. They're like a solid protein, like a thin wire. They're called microfilaments. In fact, your book talks about actually microtubules, microfilaments, and intermediate proteins that I'm not even going to deal with. Now, some of these proteins, especially the microtubules, can move. Some of these proteins, especially the microtubules, can move. And these microtubules that move are what allows movement of a cell, of what's called amoeboid movement, cytoplasmic streaming, or even contraction, changes in the shape of a cell. We're going to be, I actually brought with me a model of something containing microtubules. So these represent microtubules, tube-shaped proteins that are capable of moving. So it appears that microtubules, these proteins, are the fundamental unit of movement that allow movement of cells. We'll have more to say about that. Let's speak more about cell walls. What is a cell wall? We wrote that a cell wall is rigid but porous, and it's made up of a polysaccharide. In fact, the plant cell wall, the outer wall around plant cells that I've made reference to several times now, is made up of a polysaccharide called cellulose. We had also on page E1 mentioned that bacterial cells have an outer cell wall, so that's made up of a polysaccharide. Now, I just want to try to explain, what do I mean by rigid but porous? How can a cell wall be rigid, really strong, and yet porous, meaning things can easily go through it? Imagine this. Think of a chain link fence. Can you think of a chain link fence? Is it strong? Yes. It's difficult to knock it down. It's strong. It's made out of metal. All right? But is it porous? It's got all those openings in a chain link fence. It's easy to throw rocks right between the spaces of the chain link fence. So a cell wall made up of polysaccharides looks a lot like a chain link fence. It's rigid and strong, but very porous. It doesn't interfere with anything going in or out. All right? So just as a visual aid. Now, we wrote that the cells of most types of organisms have a cell wall. In fact, the only cells that don't have an outer rigid cell wall are animal cells, including us. But plant cells have an outer cell wall of a polysaccharide, bacterial cells, fungal cells, so most cells have an outer cell wall. Let's talk about the nucleus of a cell. Now, the word nucleus literally means what? Center. Very good. Some of you are saying, well, how would I know that? Because we told you that the center of an atom is called the nucleus, because all that the word nucleus means is center. In this case, the nucleus in a cell is referring to this large structure in the center of the cell. Now remember, of course, prokaryotic cells don't have a nucleus, such as those of bacteria. But most cells do have a nucleus. Now, so we drew a nucleus. Now, just to describe my picture here for a moment. It's an amazing picture. All right, so we have a nuclear membrane, and the structure of the nuclear membrane is very similar to that of a cell membrane. It's just like the cell membrane. The nuclear membrane is similarly made up of a phospholipid bilayer and proteins. In addition, though, we're going to be seeing in a moment that there are little pores or openings. There are pores or openings in the nuclear membrane, and we'll show you another picture that shows that. Now, inside the nucleus is a jelly-like fluid that is called nucleoplasm. And this nucleoplasm, this jelly-like fluid, is very similar to the cytoplasm jelly-like fluid here. So we've got cytoplasm here and nucleoplasm in the nucleus. All right, so cytoplasm is here, nucleoplasm is inside the nucleus. And just as we learned that the cytoplasm is mostly water and protein, 80% water and then protein, the nucleoplasm inside the nucleus is mostly water and proteins as well. Anything that's made up mostly of water and protein is jelly-like in consistency. But in addition to the water and proteins inside the nucleus, there's also DNA. Now, also in the nucleus is an area called the nucleolus. And the nucleolus, and if there's more than one, and there can be more than one nucleolus, then it's called nucleoli. That contains the chemical called RNA. So the chemical RNA is found concentrated in this area called the nucleolus, or nucleolus. Now, let me just mention something else here. And I wrote it here at the bottom. And we're going to have more to say about this. We actually talked about this subject back near the end of section D. When we were learning about DNA, we learned that the DNA molecule, or chromatid, can coil up or uncoil. And we had learned that when the DNA is uncoiled, and it kind of looks like a wet spaghetti noodle, it's said to have a chromatin shape. This is not the first time I've mentioned it. We covered it back at section D when we were learning about DNA. When the DNA coils up, when the DNA becomes coiled up, we say that it has a chromosome shape. So these terms, chromatin and chromosome, are describing whether or not the DNA molecule is uncoiled or tightly coiled up. So I would refer you back to our earlier discussion on this in section D. And I will try to get that video posted so you can rewatch the lecture. Let's take a look at page E3. And we're going to look at some pictures here that show all this much better than my pictures in just a moment. On page E3, the endoplasmic reticulum. Now the stuff that I'm covering right now is in chapter 3 of your textbook, all the way in chapter 3. Yes, we did make it past 2, but it's chapter 3. All right, the endoplasmic reticulum. Now what does the word literally mean? Endo means on the inside. Plasmic means cytoplasm, so on the inside of the cytoplasm. And reticulum means a tubular network. A tubular network. So there is a tubular network, a reticulum, inside the cytoplasm. The way that I wrote it is that there's a network of tubes or tubular canals that connect the nuclear membrane with the cell membrane. And it functions like a little circulatory system inside the cell for the transport of molecules. Now, just to help you visualize this, why don't you look at page E2II. That's this page. Now, it came with the lecture outline, although I gave you one last time. We passed it out to you, and it said B2II, same thing. All right, so this is showing a cell. Here's the nucleus, and you can see there's this tubular network. This tubular network, this network of tubes inside the cytoplasm, is called the ER, endoplasmic reticulum. Reticulum means a tubes, tubular network, endoplasmic, inside the cytoplasm. Now, there's actually two types of ER, or endoplasmic reticulum, two types. There's what's called smooth ER, and what is called granular, or rough ER. Now, we'll have better pictures of this in a moment, but you've all got this picture. All right, now, the smooth ER is called smooth because it's smooth. You'll notice how it's attached to a series of flat sacs. They look like pancakes. And this series of flat sacs that look like pancakes is called the Golgi Complex, named after a cell biologist, an Italian cell biologist named Emilio Golgi. Now, the importance of the smooth ER. Well, first of all, the endoplasmic reticulum in general, this network of tubes, acts like a little circulatory system inside the cell. Just as we have blood vessels that allow substances to be transported throughout our body, at the microscopic level inside cells is essentially something that's like blood vessels called the ER. It's a network of tubes that allows chemicals to be transported from one part of the cell to another. It's just amazing the complexity at the level of a single microscopic cell. Now, in addition to the role of these tubes allowing chemicals to be transported from one part of the cell to another, they have another role. The smooth ER is involved in lipid metabolism. You'd say, what? It's involved in the manufacturing and breakdown of fats. Lipid metabolism. So this is where lipids, fats, can be manufactured, synthesized, manufactured, or even broken down. For an example of a fat that is manufactured in the smooth ER are steroid hormones. Because we've learned that steroid hormones are actually chemicals, hormones made from what fat? What are steroid hormones made from? Which fat? Cholesterol. So that was back in section D, page D3. So anyhow, cholesterol is used to manufacture steroid hormones in the smooth ER. Now, there's a second type of ER called granular or rough ER. And it's called granular rough ER because it's got little granules associated with it. Now these little granules are called ribosomes. And these little granules called ribosomes are right next to this tube. Notice again how this tube is attached to a series of flat sacs that look like pancakes, again known as the Golgi Complex. Now, the purpose of the ribosomes of what's known as the granular or rough ER is that proteins are manufactured. Proteins are manufactured or synthesized from amino acids at the ribosomes of the granular ER. So now we're telling you where in a cell steroid hormones are made and where in a cell proteins are made. So we've gone beyond knowing what steroid hormones are and what proteins are. Now we're telling you where in a cell these different chemicals are made. Okay, so that's why you got to make sure you know what all those chemical names are, because now we're going to go into the cellular level. Question?
All right, you said that the smooth ER makes them?
Yes, it makes them and even breaks them down. It manufactures and breaks down steroid hormones and other fats. All right? And proteins are made or broken down, well, let's say made, in the ribosomes of the rough ER. All right, so that's kind of gives us an idea of this. Let's look at page E3 again, because we wrote all this. All of this is written on page E3. On page E3, in addition to saying that the ER acts like a little circulatory system for the transport of molecules, we wrote that smooth ER is involved in lipid and steroid metabolism, such as the manufacture of steroid hormones. The rough or granular ER, we wrote, is studded with, that means it's got little small bodies called ribosomes, and these ribosomes, which are made up of RNA and synthesized in the nucleolus, the ribosomes are where proteins are synthesized. That's where proteins are made. Let's say a little bit more about the Golgi Complex. Now the Golgi Complex, and some books call it the Golgi body, some books call it the Golgi apparatus, I've called it the Golgi Complex. It's called Golgi in all these cases, named after the cell biologist who first discovered it. An Italian biologist, you certainly don't have to know his name, but his name was Emilio Golgi. What is it? It's a stack of sacs. I said it kind of looks like a stack of pancakes. The purpose of those sacs is that's where these chemicals can be stored. So the Golgi complex is where various chemicals are stored and modified. The various chemicals that were formed in the ER. So both proteins made in the rough ER and steroid hormones made in the smooth ER can be temporarily stored in these flat sacs of the Golgi complex. Let me go just a drop further and then we'll try to put this story together. Now, what's listed next on Roman numeral 10 are storage vacuoles and vesicles. You'd say that again. Vacuole and vesicle. These are sacs. And if the sacs are large, these are sacs for storing or containing chemicals. If they are large in size, they're called a vacuole. If they're small, they're called a vesicle. All right? And so we've listed a number of these vacuoles or vesicles, and in fact, the first one I've listed, and I wrote it to your right, are secretion vesicles. All right? And so now I want to try, before I go any further and read anything more here, I want to try to put this story together. So let's look at page E10. E10. It says E10 right here. We're going to look at page E10. This is page E10 at the top. This is a picture of a typical animal cell. In fact, both the top and bottom pictures on page E10 are typical animal cells, or including human cells. And you'll notice, let's see what we can identify. First of all, remember you've got pictures in brilliant color in your textbook of all these cells and all the parts of a cell in chapter 3. But you can see it points to, on the right, the cell membrane, the mitochondria, microtubules, the Golgi body, the nucleus. It goes on and on and on. And these are shown in, even nicer in your book. But I want to draw your attention to a few things with this upper picture. First, this is the nucleus. And it's labeled that, nucleus, on the right. You'll notice that there's a nuclear membrane, and on the left-hand side you can see there are holes or openings in the nuclear membrane. These are called pores or openings through which chemicals can go in or out of the nucleus. You'll notice that inside the nucleus is a structure called the nucleolus or nucleolus, and that contains the chemical RNA. Now there's also this jelly-like fluid inside the nucleus called nucleoplasm. So the jelly-like fluid inside the nucleus is called nucleoplasm, and I just wrote that in there. You can too. And we've already mentioned nucleoplasm, like cytoplasm, is mostly water and proteins, having the consistency of jello. In addition, there's DNA in the nucleoplasm. And in fact, you'll notice that it has a label on your picture that says chromatin. You'd say, yeah, what is that? That's the name that we give to DNA when it's uncoiled. So in fact, all right, so this is page E11, and this is a cell right here. And it's showing what can occur inside this cell. This in the form of a flow chart. Now, if you're not familiar with flow charts, they use flow charts for all kinds of subjects nowadays. Flow charts are basically made up of boxes and arrows. And the purpose of the boxes and arrows is to show you a sequence of events. What happens first, what happens second, what happens next. So let's just read what it says in this top left box. It says in the top left box, ribosomes synthesize proteins, right, in the rough endoplasmic reticulum. And these proteins include enzymes. They include, mucus. They include hormones, protein hormones, and antibodies. All right, isn't that what we've learned about the ribosomes of the granular ER? Now, let's read what it says in the box right below it. The smooth endoplasmic reticulum synthesizes and stores lipids. These lipids include steroid hormones. Isn't that what we learned about the smooth ER? Now, you'll notice that both boxes, the upper one and the lower one, have arrows pointing here. Does everybody see that? It's pointing to the middle box of the second column. All right, so what can happen next after either proteins are made in the ribosomes of the rough ER or steroid hormones are made in the smooth ER? What can happen next is that the proteins or lipids can be transferred to the Golgi complex. Now the complex, the Golgi complex, sometimes joins carbohydrates to the proteins or to the lipids. So if you were to join a carbohydrate, carbohydrates are sugars, if you joined a carbohydrate or sugar to a protein, that would be called a glycoprotein. And we've learned that recognition sites are glycoproteins, for example. So the flat pancake-like sacs of the Golgi complex not only store chemicals, but can modify them as well. Now you'll notice that there's an arrow pointing to the right. So what may happen next is shown in the bottom box of the third column. So what might happen next is that proteins or lipids can be transferred to secretory vesicles, and these vesicles can fuse to the plasma membrane and release their contents to the outside of the cell. And what do we call it when something is released to the outside of the cell? Secretion or exocytosis. Is that pretty much exactly what we've said can happen? All right, so let's give you 3 examples of this. 3 examples. I just wrote them right here in this kind of blank space, right on page E11. So, example number 1, pancreas cells that secrete insulin. First of all, have we ever learned previously that insulin is made by pancreas cells? Yes, we did, back on page D5. All right, so let's imagine we were looking at a pancreas cell through the microscope. You'd say, I can't imagine that. Okay, let's help you. Turn back to E10. Let's go back to E10. Let's say that this picture on the bottom of E10 is a pancreas cell that makes insulin. Where would the insulin be made in this pancreas cell? At the ribosomes of the rough ER, because insulin is a protein. It's a protein hormone. And where would this insulin be temporarily stored in this pancreas cell? Very good, in the Golgi complex.